process monitoring. The guideline also emphasizes several things, and one of the things is air visualization studies, where the laminarity is demonstrated. Also, when the return air goes, it should your HEPA filter or air velocity should be such that it gives a laminar airflow, as well as it is sweeping the air and the air changes are getting maintained. In my experience, I have seen that most of the companies which are doing air visualization studies, and for that they are taking the help of outside people, and they don't look into whether this is done in a right manner or not. Similarly, there is a system called media fill, which is a sterility assurance. In the today's guideline, we have 5 clauses, whereas this has been expanded to 19 clauses. So lot of requirement has been emphasized. Also, in the draft guideline, the utilities has been given a lot of emphasis, like water system and steam. In the case of water system, there are numerous references has been given how to mitigate the biofilm formation, because when the water is flowing, there is a chance of biofilm formation, and the water should always be maintained at a turbulent flow and at high temperature. Similarly, the new thing which has come is the water for injection you can produce by non-distillation method. That is why RO is also acceptable. Earlier it was not there, and there are other. Due to time constraint, I cannot take everything. So in the end, as a summary and conclusion, I can very well say that Annexure 1 draft 2020 is a rewrite and not a simple revision. The 2008 contains about 127 clauses, whereas the new draft has a 300 clauses. So it is more than double, which is a very high level of detail is provided. And in many sections, you can find a roadmap of how you can implement, how you can make a robust SOPs. So it is very clear that the Annexure 1 draft that regulators are expecting when they are on the manufacturer's side, that to really understand their process in entirety, and they can then demonstrate that they have adequate contamination control in place, and they have risk mitigation plan, and they have a effective documentation, whether you have a electronic documentation or a paper, you can demonstrate that everything is under control. This consultation process is still going on, and a final document is not yet published. So this can be taken as a reference document to improve your systems, and this cannot be taken as a hard rule. If any company has any alternate approach, they can do so. So I have made my presentation based on a few of these reading materials, and I want to acknowledge those. So if any of you want to get in touch with me, now I am a freelance consulting company, so I have provided my email address and my website is there. So you can approach me if you have any questions in this regard. So thank you very much for the patient listening, and now I will stop my presentation. Thank you very much.
Thank you, Dr. Mishra. Now the session is open for any questions. They can ask the question.
Dr. Mishra, thank you very much for a nice presentation. But I have 2 questions. I'll ask one by one. First question is, there are significant differences as we can see from the FDA perspective as well as European perspective now. So how the companies or industry, Indian industry should adapt to it?
European perspective and what you asked?
FDA.
US FDA.
Yeah.
No. This is my view, Mr. Jadhav. Earlier, when I started my career 20 years back and 30 years back, there were perspective difference between US FDA and European approach. But slowly, now there is no difference, and hardly there is any. If you can give me any example, then I can comment on, because I have worked for 30 years in injectable plants also. In injectable APIs also, non-sterile API, oral dosage form and everything. So I can share my experiences. But what is happening now, if you look at even the 2008 EU Annex 1 and the European, the US FDA injectable guideline of, I think, 2004, both of them are same. Earlier there were some difference of, you say like in Europe, they call it in the pound, and somewhere we did a kg. So earlier the environmental monitoring, it was shown as a square feet and a square meter, something like that. And the air you are sucking, square meter, square feet. Those kind of differences were there. Are class 100, and in ISO system it is class A, B, C, D, something like that. So nowadays, most of these guidelines are harmonized, and this EU GMP and European, I don't think there is any perspective difference existing as of now. If you give some very specific example, I can dwell upon, but as on date in 2020, just don't have any. And one more thing, I want to just add here, if I am not elaborating my time, the guidelines are minimum requirement. Everything you cannot, even if this is a 52-page guideline, you cannot write everything. So what the industry is expecting from what you call inspectors are expecting from the industry is, you should also go beyond what is the industry practices, what is the technology advancement taking place, what is written in other guidance document like WHO, ISO, like in air quality and other thing, ISO 14644, 1, 2, 3, 4, these all are, and these references are there. Then also this PDA, ISPE. Actually, when these guidelines are coming, these people are comprising the consultating committee, and at different levels they give their inputs, and also the advancement of science and technology is playing a greater role in revising these documents. So to conclude, there is no difference between US and Europe perspective, very minor, maybe about 1% or so, and it is actually the inspector's perspective, because you have 100 inspectors, so each inspector's background is playing an important role, so how he is interpreting, but more or less it is now harmonized.
Yeah. No, with respect to detailing also, like number of clauses have increased almost.
Yes. Yes.
That's what. But then will not Indian generic industry be hard pressed, because already it is struggling with respect to US FDA guidelines and other guidelines? We will have infrastructure wise and the compliance wise, you can say.
See, there are many technical things I can talk, then there are many non-technical or other issues which I cannot talk here. As you are aware, like you can say, we were in India, Mr. Gaikwad, I have 5 minutes more, or I have to conclude?
Yes, sir, we'll take this question and then.
Yeah. So, like in India, you see, we were the world champion of hockey. What happened to that world championship consistency? Because rule has changed. From the grass, it is turf. So what happens, India is a great threat. It is not a threat, but it is a great challenge to the global world of producing low cost quality products. So what happens when you enter in their domain, they will come out with a very high level of compliance involving sophisticated technology. And that's where India will struggle. I will just give you one example, like a tableting machine, or even you take an example of a lyophilizer. The 1 liter lyophilizer will come about 27 crore automatic unloading and loading. Whereas in India, you can get a lyophilizer in 5 to 6 crore. So you see the difference. Certainly the 27 crore lyophilizer will give you much more comfort, much more precise everything, which is very compared, Mercedes and Honda. So those things are there, but India is overcoming, and technology and the software system will certainly do that. So thank you very much, and we can discuss separately on this topic, and time is not allowing. So I'm also sorry and apologize for extending my time for about 5 minutes. I could have finished about 5 minutes earlier.
No problem.
Thank you for that. Thank you.
Thank you, Dr. Mishra, for your wonderful talk on the highlighting the role of GMP and sharing your views on the European guidelines, which is the draft guideline on 2020. Thank you very much. Now,
Thank you.
Thank you. Moving to the next speaker, who is there, Dr. Ajit Nair. So it's my pleasure to introduce him. Dr. Ajit leads clinical development for Ryzen Pharmaceuticals and is currently Senior Vice President and Head Clinical Research Development. Dr. Ajit has around 24 years of experience in clinical research and development and medical affairs, and has held senior leadership position in companies such as Novartis, Glenmark, and Zero Clean Pharma. Dr. Ajit has lead teams of clinical development professionals through the entire government of research activities, including clinical development, science and strategy, clinical operations, and clinical data sciences. His experience covers oncology, metabolism, and immunology drug development. He has spoken and presented at multiple seminars, workshops on clinical development, and has presented data at multiple international conferences. He is alumnus of Bombay College of Pharmacy, Mumbai, and has a PhD in pharmacology. He is an avid marathoner and is passionate about bridging industry and academia. So, thank you, Dr. Ajit. Now I'm handing it over to you for your talk.
Okay, thanks, Mr. Gaikwad, and thanks to everybody, the faculty of pharmacy at BITS Pilani, to give me this opportunity to discuss issues around regulatory, around the regulatory landscape. And I'd like to focus my talk on a different topic, which is probably a bit different from what the other speakers have been talking about. And this is more around clinical development and the regulatory perspective of clinical development for molecular entities in oncology. So I'm going to talk a bit about the overall landscape of how regulations in the oncology development space for new molecular entities have sort of been framed and reframed and repositioned so that access of medications to patients who need it most, like patients with terminal advanced cancers, get access to medications quickly, rather than get stuck into the whole loop of bureaucratic procedures, processes, and regulations which can delay the drug coming from the lab to the clinic. So I'm going to talk more in general about these aspects, and then what should be a best clinical come regulatory strategy to take your compound from the lab in discovery to the clinic in the patient. So I'm going to talk also a bit about that strategy, which will help the drug to reach the market faster. So I'll start sharing my screen. So Mr. Gaikwad, just let me know once you're able to see the screen.
Sure, sir.
So are you able to see my screen?
Not yet.
Not yet. Okay.
So So on the bottom right, you will have present now option.
Yeah, that is what I'm looking at.
Your file has to be open for that.
Yeah, I guess you can see it now.
Yeah, it's coming now.
Okay, great. Right. So this is the title of my talk, Developing an Optimal Clinical Regulatory Strategy in Oncology Drug Development. And I'm going to focus more around oncology drug development in the new molecular, the new chemical entity space. I'm not going to cover anything on generics or biosimilars, which is a separate discussion topic altogether. But this is more around NC development in oncology. So I just give you a brief idea of the overall space landscape of cancer and oncology at a larger level. So, and this is more to give you statistics and to give you an idea of the issue and the problem we have in terms of the disease oncology, right? So this is the 2018 Globocan data, which talks about the spread of cancer across multiple continents in different parts of the world. And at any point in time in 2018, there was about 18 million cases of new cancers detected in the world, right? And if you look at the split of cancer cases across different continents, you can see that a major part of that is occupied by Asia and Europe, where the issue or the burden of oncology and the burden of advanced intractable cancer is pretty high. So India also, I'm not going to go into the breakdown of Asia into India and the other countries within Asia, but India has a very large burden of cancers coming up, especially in areas of head and neck cancer, breast, ovarian cancers. The numbers are really going very high. And if you really take a snapshot of this 18.1 million new cases, which you see, you basically are also seeing a lot of these cases not really being treated very well. So half of this population actually dies every year. So in 2018, or the 18 million cases of cancers detected, the same year there were about 9 million patients who died from advanced cancer. So it's going to be a big global problem as a number of cancers increase globally. And you would have seen, and there's a lot of focus now in terms of proactive cancer testing in patients really looking at advanced treatments for the disease, physicians and the general public being made more aware about cancer programs and how to prevent cancer. There are a lot of insurance companies which have specific cancer insurance coming in because there is an increasing trend of cancer really overtaking cardiovascular disease in terms of being the leading cause of death and morbidity when it comes to patients with the disease. So we're looking at really a big global problem of the incidence of cancers going up. And just to share a perspective of why this is happening, it's happening because of a lot of issues, right? And the issues are around the disease itself. And this is a iconic figure called the hallmarks of cancer. And if you look at this whole figure, it really gives you a perspective of how cancer is such a difficult condition to treat. And if you look at these hallmarks, these are basically 10 different mechanisms by which cancer cells grow. Cancer cells sustain their growth and eventually kill the host, right? So here is what is a picture called as hallmarks of cancer, which talks about different mechanisms by which the tumor cells will either grow rapidly, they will evade immune suppression by the active immunological functions of the body, they will invade other tissues by active metastasis. There is a lot of process where they can really increase a lot of blood supply to the tissue. There will be a lot of increased metabolism, which allows more glucose and other metabolites to come in to help the tumor to grow. So it's basically a multifaceted disease. And if you target one aspect of this hallmark, the other aspect gets propagated. If you block 2 or 3 of them, another pathway grows. So it's technically a very complex disease. And if you really look at drug development in cancer, why is cancer not really still considered a 100% curable disease? The reason behind that is if you block 1 or 2 pathways, there are 7, 8 pathways still remaining, and you don't have a drug which really blocks all pathways, right? So cancer cells are so very smart that they can use alternate pathways, alternate mechanisms. You block one mechanism, another mechanism propagates. If you block that mechanism, a new mechanism comes into place. So cancer biology, as you know, is extremely complex, right? And this is the 10 hallmarks of cancer which we see now. But there could be several other mechanisms which are still unknown to humans, where cancer cells could be using propagating. As you would know, the Nobel Prize for Cancer 2 years ago went to MD Anderson Cancer Center, where they really looked at something called an immune checkpoint sort of mechanism, which was being used by cancer cells to evade the immunological attack by the normal immune cells, and that led to a lot of drugs. For example, Merck's pembrolizumab or Keytruda, Bristol Myers Squibb's drug, which really works on these mechanisms, right? So the more we understand the biology, the more complex it becomes. And it's fascinating that these biologies are actually being used in drug development, because a lot of these mechanisms and the targets which are used by these mechanisms are druggable targets, right? So a lot of these mechanisms can be used to make drugs or molecular entities which can attack to one mechanism. And if that mechanism is the most prominent mechanism, you potentially can achieve cancer arrest. So this is the perspective of the hallmarks of cancer. Moving to the next slide, again, it's more of a slide which talks about the complexities of oncology drug development. And like I just showed you in the previous slide, the biology is extremely complex, right? I basically consider oncology as a disease which is like software, right? You have some software today, it can become redundant tomorrow, right? You have a certain technological advancement today in information technology, it can be overridden in 2 years. In a similar way, you think you've addressed the whole cancer biology very well. You think that you've created all possible drugs, and the cancer tumor cell will now evade that, come up with a new pathophysiological mechanism to grow, and it starts growing. So it's such a complex disease that you think you've conquered it today, but you've actually not conquered it. So that's the complexity of the disease, right? And because now the understanding of cancer biology has become so very, very comprehensive, that a lot of targets now can be druggable, which means that you really can discover and develop drugs on targets which were never known before. And now, since they are known, you really can target those areas where you can develop drugs and therefore suppress tumor growth, right? So that's the evolving landscape which happens, and we're looking now at an era, and it's significantly moved from the last. If you really look at cancer therapy 20 years ago and cancer therapy today, right? And I'm talking from an overall global perspective, not specific to India, because unfortunately, a lot of these cutting-edge technologies and cutting-edge medications have not reached India from a commercial and a patient access perspective. But talking about from a global perspective, a lot of regimens now, which earlier used to be 100% chemotherapy based, have actually shifted to target therapy based. So there's no longer chemotherapy given in certain indications where the therapy is completely chemo free, and that really is a paradigm shift in the way cancer therapy was 20 years ago versus what cancer therapy is now. So it's really advanced, but in spite of all these advances, it still remains a highly unmet medical need in terms of the disease, right? So there's a lot of targeted therapies and personalized medicine which are being developed. So you basically can now look at medicines being developed which specifically attack certain mutations, right? So as you know, I mean, the cancer as a disease is a highly genomic unstable disease, which means that there's a lot of genomic instability, there's a lot of mutation which happening, there's a lot of DNA repair, break, and all that stuff happening within a tumor cell, right? So you really can look at some tumor cells which express a certain type of gene mutation, or you can look at a certain tumor type which expresses or overexpresses a certain protein kinase, right? So you can look at some of these areas where there's overexpression of a target, or there's overexpression of a protein, there's overexpression of a certain area which allows the cancer cells to grow. And therefore, you can basically identify what is the mutation or what is the overexpressed protein, and you can specifically have the patient receive only that therapy which can work on that overexpressed protein or overexpressed substance, right? So that is how medicine is becoming more and more personalized in cancer. And of course, some of you must, or many of us must have read about the latest CAR therapy, right? Cell-based therapies, the CAR therapies hold immense potential, and that's really going to the personalized level where you basically have the patient's own blood cells being used, repurposed, pushed back to the patient to really kill the tumor, right? So this is again the cutting edge work which is happening in the field of oncology, especially on cell-based therapies, CAR therapies. However, I mean, like I said, there are still cancer as a disease really is a recalcitrant disease, especially in some indications where we still don't have effective therapies, right? And the overall survival can be really, really very low, right? So some of the diseases, for example, a disease called peripheral T-cell lymphoma, I mean, the overall survival at the end of 5 years is less than 30%, which means that if you have 100 patients to start off with today, at the end of 5 years, there'll be 70 patients who are dead, in spite of whatever therapy you give, right? And only 30 patients would still be surviving. So you have some diseases which really are very tough to treat. They're still not effective therapies, and some of them are really orphan diseases where there's not much of drug development happening, right? So therefore, I mean, it's very important for the industry and the regulators to really work very close to each other, see that drug development is done properly by the industry. At the same time, the regulations are not so bureaucratic or not so tight. And at the same time, there's an out-of-the-box thinking which regulators can do so that medications don't have to run through the standard approval process, but that they can be a sort of out-of-the-box way of looking at things when it comes to getting cancer drugs approved quickly so that they eventually reach the patient who needs it much more faster. As against other conditions like diabetes, hypertension, where they may follow a standard pattern of regulatory review and approval, whereas for oncology indications with difficult-to-treat diseases, you obviously need something which is path-breaking. And I'm going to talk about that. Coming to the IND, and as many of you know, IND is something called as an investigational new drug application. And I'm talking more in relationship to the US FDA, and I'll talk more about that because the US FDA probably is one of the agencies in the world which has a very broad, forward-looking thinking in terms of regulations, especially when it comes to oncology development, right? So when we classically compare this with some of the non-oncology INDs and an oncology IND, there are some stark differences in the IND requirements for filing with the FDA for an oncology drug versus a non-oncology drug. Some of you must be knowing there are certain requirements from an IND perspective. The requirements would be around your basic in vitro pharmacology. You need toxicology data, right? Especially a 28-day toxicology data in a rodent and a non-rodent species. You need full chemistry manufacturing control information, specification specs on drug substance and drug product, right? You need all the requirements which go into a classical IND. And of course, your phase 1 protocol, which is to be tested in humans. So that's the complete IND package, which is required when you file any product with the FDA for a first-in-man study, right? And these are some of the differences which you see primarily from an oncology product versus a non-oncology product. There's something called a safety pharmacology, which is part of the package which goes in the non-clinical sections of the IND. And the safety pharmacology is nothing but testing which is done in an animal species to understand the effect of the drug on the cardiovascular system, on the nervous system, and the respiratory system. Now, these tests basically are required as part of your standard tox studies which go for a non-oncology product that is actually not required or waived off for an oncology indication. Then you need genotoxicity data and reproductive toxicity data for a non-oncology product, which is not essential at an IND stage for an oncology compound, right? So that's again a key difference because all this adds to time. So your IND can actually be put together much more quicker for an oncology IND as compared to a non-oncology IND. Very important point is the third bullet, which is a 4-week tox. So essentially, as you would know, a tox program is a very important program which actually mirrors a clinical development program. You can expose humans to only that much duration of your investigation product as much as tox data you have. So for example, if you have a 28-day rodent, non-rodent toxicology data, you can expose humans only for that period, right? So if you have a 28-day tox, you can give the medication or the investigation product in the humans only for 28 days. If you have 3-month tox data, you can expose human beings 3 months of continuous dosing. If you have 9 to 12 months of data, you can expose human beings 1 year, right? So those are the regulations which are typically there for a non-oncology IND. There is a difference in the oncology IND. If you do a 28-day tox in a rodent, non-rodent species, pretty much that 28-day tox can really cover 1 to 2 to 3 years of human exposure without the need of long-term tox to support long-term human exposure. So that is what any regulation, this is not just the FDA, even for the EMA, the regulation is the same. You basically need just 28-day tox to really cover human exposure much beyond 28 days, which can go into years together, right? But the only requirement is that if your drug really works and you have positive efficacy and safety data, When you file for an IND, you need to submit a 3-month tox at that time when filing the IND. But the 3-month tox is not required during your development phase. A 28-day tox will be enough during a development phase, right up to a 2 or 3 year exposure to really support the IND. So that's one key stark difference for an oncology IND from a non-oncology IND. And of course, as you would know, there are chances that your investigation product may have metabolites, and the formation of metabolites can vary across species, right? So you may see a metabolite of your parent compound not seen in a rodent species or a non-rodent species, but you probably see a metabolite coming up in the humans, right? So generally, from a regulatory perspective for non-oncology INDs, you really have to characterize and qualify that metabolite in a non-clinical species, whereas that is really not required and waived off for an oncology indication. Now, the question which will come in is that why are these things being waived off oncology? I mean, why for non-oncology all these things are required, whereas for oncology it's required? Now, remember, oncology is a complex disease. Oncology is a difficult to treat disease, and the overall survival in oncology is pretty low, right? So for oncology as a special indication, any regulator will waive off some things because these things take time. And these things in an oncology setting doesn't matter because oncology per se is a difficult to treat disease with very, very compromised overall survival rates. So therefore, the FDA or the EMA has a more lenient view on some of the things which are really required for oncology products, but it's really waived off oncology compounds. So that's one of the key stark differences of an IND for an oncology versus a non-oncology application. Now, I'm going to talk a bit about the study designs and relate this to the regulatory aspect. And if you really look at study designs in oncology, and this is I'm talking about phase 1 study designs, there are basically 2 types of broad-based study designs. One are called as umbrella trials, and the other one is called as a basket trial. And I've highlighted the one in a basket trial primarily because that's trial which most industry groups will do because they're looking at developing a particular drug. Whereas the umbrella trials are run by the National Cancer Institutes or by big cancer centers like MD Anderson Cancer Centers. So it's more investigator-driven trials are the umbrella trials, whereas most of the trials the industry runs are the basket trials. And if you look at the basket trials, the essential aspect of a basket trial is that you basically have a drug, right, which particularly targets a certain protein or a certain enzyme, right? And this drug basically will target multiple cancer types in which this expression of the protein is abnormal. For example, cancer A, B, C, or D, you primarily will evaluate your drug in all these types of cancers which show an overexpression of that particular protein, right? So you basically, from the beginning itself, are able to test your drug in multiple tumor types A, B, C, and D. And if you see a signal of efficacy in a certain tumor type, you will pick that signal and you will amplify that signal going forward in the phase 2, phase 3 design. So the design primarily at a phase 1 level is to pick and choose the right tumor type which is reacting to your drug, because that particular overexpression of protein or a mutation, right, or an aberrant signaling pathway could be prevalent in an A, B, C, or D tumor type, but it is probably more prevalent in, say, tumor type A or tumor type C. And if you see your drug working in that particular tumor type, you will basically take that tumor type for further phase 2 development. So this is the overall concept of how we really work with a phase 1 design in oncology. And all these phase 1 designs, mind you, are vetted by the FDA and the EMA. So the EMA also has a common guidance. So there are a lot of guidances if you look at the FDA website on oncology development or the EMA website, it talks about early phase development, clinical development in oncology. And some of these designs are right there, right? So you can really look at some of these designs and make a strategy, right, based upon these designs, which has also been proposed by the FDA. And again, to give you a broad-based idea of how the designs look like, and just to contrast this with the typical designs which you do in a non-oncology setting. I mean, remember, oncology compounds, obviously you cannot do oncology phase 1 designs in healthy volunteers, whereas all non-oncology indications like hypertension, diabetes, arthritis, asthma, all these compounds in a phase 1 level will always be done in healthy volunteers, whereas all oncology designs will always be done in patients with the target disease, right? So remember, all phase 1 designs, you have patients with advanced cancer. So when you look at phase 1 designs, typically there are multiple designs, and probably this talk is not the right forum because it's out of the scope of this discussion. But the designs can be a standard 3+3 design, they can be a Bayesian design, they can be a rapid titration design. But the design here primarily talks about a dose escalation phase. And as you would see, I mean, we're jacking up the dose from an XMG to a 2x to a 4x to an 8x mg, and that XMG is basically decided from your tox studies. Your tox study decides that XMG dose, and the XMG dose is escalated till you find a dose which is tolerated by the patient, right? So if you see that 8x milligram here probably caused extreme neutropenia, right? The patient's WBC count really, or the patient's neutrophil count really dipped massively, right? And the patient needed a dose of GM-CSF. That dose of 8 milligram is supposed to be a dose-limiting toxicity. And therefore, the dose below that, which is 4 mg, becomes your maximum tolerated dose. So the 4 mg dose, primarily the dose which should be taken for the next level of experimentation, right? So, and that's called as the MTD dose, the maximum tolerated dose. And this is a very standard terminology in phase 1 designs in oncology. So the 4 mg dose, primarily after the dose escalation phase, is taken into a dose expansion phase. And here it is where the basket design comes in. So I have a tumor type A, I have a tumor type B, I can have tumor type C, I can have tumor type D. I will use the MTD dose, which is the safe dose, to see whether that safe dose now gives me an efficacy signal in a tumor type A, B, or a C, right? And that is how I do my development design based upon the basket design, which you saw on the previous slide. Oncology development as such, and from a regulatory perspective also, generally breaks all rules, right? So, and like I said, oncology is a special indication because the type of patients you're dealing with are terminally ill patients, right? And for which you really cannot follow standard rules. You have to break the rules, right? So one of the key things where oncology breaks the rules is challenging the gold standard. And if you know, I mean, the gold standard for all clinical trials is randomized, double-blind, active stroke placebo-controlled studies, right? So that's the gold standard study because that's a study which a regulator accepts for all non-oncology designs, where you really have to have a study which is randomized and double-blind, right? So that's the gold standard. But with oncology, you can really challenge that design, right? You can really look at single-arm open-label studies which can really be filed for registration. So if you look at the US FDA, and that is why I was saying the US FDA is the most progressive in this respect. So the US FDA is amenable and open to look at trials which are not randomized but a single arm, which are not blinded but are open label studies, right? And you can use that data, right? And use that data to really file if your drug really works there. So that data in a single arm open label study can be used for registration purpose with the FDA. So that broad base is the FDA thinking when it comes to oncology development. You can also look at retrospective controls instead of prospective controls, right? So when you do randomized double-blind comparative study, you have to have a prospective active control, and that active control could be a placebo or the active control could be the standard of care, right? Whereas in oncology, you can actually use a retrospective control. You can have a single arm study, but the second arm of that study is actually invisible arm, which is actually historical data from a positive control, right? So you have the liberty of using a retrospective control instead of a prospective arm in a randomized study in oncology. And that's the beauty of how you can really play around with oncology designs, because the FDA or the EMA is very open to such innovative designs which allow retrospective controls also. But mind you, remember, this is very important only in those cancers which are really intractable, difficult to treat, where really there's no much of drug development happening. And I'll come to more special cases in the subsequent slides. So retrospective controls can also be used. The other important aspect is, I mean, if you remember the slide which you talked about the expansion cohort. In the expansion cohort, if I see tumor type A, tumor type B, tumor type C, and tumor type D, or which if tumor type C shows me an efficacy signal, I can really drop all the other tumor types, take the tumor type C, add more patient numbers to that so that it's statistically significant, and take that tumor line straight for registration, right? So a lot of times, a phase 1's expansion cohort can be a registration directed study, which classically again breaks all the rules, Because the rule is do phase 1, do phase 2, do phase 3, and then file for application, right? Whereas in an oncology setting, in a highly unmet medical need indication, your phase 1 expansion design itself can be extrapolated and taken towards registration. That's the beauty of the design in oncology settings where you really have a highly unmet medical need, right? So that's the whole picture, right? And that's what I'm saying. You can really merge all studies. Your phase 2 can be merged with phase 3. It can become one phase 2 registration directed studies. So that's how most of the rules do not apply for an oncology design, and primarily the fact that you need patient access faster for innovative medications in oncology. This is another interesting concept. It's around surrogate endpoints, right? And when we look at endpoints, obviously endpoints, like all of you know, is obviously the marker of response, right? You really have to look at what is the marker of response to say that my drug A is working against placebo B, or my drug A is better than comparator B, or my drug is similar to comparator B. Now, in oncology, right, it again gives you a waiver. It looks at endpoints which are not classical endpoints, but surrogate endpoints, right? The classical endpoint in an oncology study is how long is the patient living, right? Death can be an endpoint, which is overall survival. How long does the patient, after taking an oncology investigation product, lives, right? That's an overall survival timeline. So the classical endpoint always has been overall survival, but the FDA or the EMA is now willing to look at surrogates. And surrogate are not necessarily the traditional endpoints, but they are a reflection of the overall endpoint of overall survival. So they are used in lieu for the classical endpoint. And some of these surrogates are called as objective in oncology, objective response rate, complete response rate, progression-free survival, event-free survival. In some of the hematological malignancies, the blood cancers, something called as major hematologic or cytogenetic responses. So these are again indicators that the regulators are willing to look at surrogates and not the traditional endpoints. So for example, I mean, if you're looking at a diabetes study, you will have to show HbA1c reduction to that certain extent. And from a safety perspective, you need to expose minimum 5,000 patients in a diabetes study to really get an approval. Here, I can look at a 100-patient study, or in some cases, a 60-patient study, which can give me a registration with a surrogate endpoint like a complete response rate or an objective response rate. So oncology looks at things differently from an endpoint perspective, from a patient number perspective, because the need of the hour is to really expedite product development or clinical development and take the compound to the market and the patient much more faster, right? So these are some of the surrogates. You can probably browse the web, and I mean, I can talk about this at length, but it will go beyond the discussion time for the presentation. But these are some of the endpoints which are really surrogate endpoints in classical, as against classical oncology endpoint, which is overall survival. Again, something which is done by the FDA here, and again, the FDA, like I said, has been the most progressive agency when it comes to oncology drug development, right? Their task essentially is to really see how quickly can you get life-saving drugs with a very good risk to benefit ratio to the patient quickly, because the patient is already in a very bad shape. How quickly can you get these drugs to the patient, right? Either a patient who's terminally ill or a patient who's been diagnosed, but you know that disease will rapidly progress in terms of disease progression and can eventually kill the patient, right? So this is where the regulator looks at things where you can expedite drug development, right? There are designations which are given to drugs in development. Well, it's fast track designation, breakthrough designation, accelerated approval, and priority reviews. Now, all these essentially are put in place, and this is not just for oncology. I mean, there are some indications like epilepsy. There's some indications like, for example, Alzheimer's and some of the other cardiovascular conditions, right, where some of these designations still apply. But most of the pharma companies use these designations primarily in oncology development because they really, really support expedited review and development. So just to give an example, I mean, fast track designation is primarily given to companies who are developing drugs in areas where there's a highly unmet medical need. So for example, in a condition of a certain cancer, the there are some drugs available, but they're not really working well. The patients are progressing very fast. The patient at stage 1 gets to stage 4 of the disease very quickly. The overall survival is low. There is extreme side effects of the existing therapy in terms of infections, in terms of neutropenia, in terms of liver toxicity. So a fast track designation primarily is given to a compound which basically addresses an unmet medical need in a particular indication where existing therapies are not really working well, or there's a side effect or a therapy which really is not being tolerated well by the patient, right? So that's how you get a fast track designation, because your drug, the early clinical data, and mind you that you cannot get a fast track designation till you have clinical data. So all these will require some amount of clinical data first to show that it is working better than what is there in the market. Right? So once you have your early clinical data coming through in the phase 1 setting, you can actually apply for a fast track designation with the FDA and say that, listen, I have a drug product here. Although it's early development, I have a potential where it is better in terms of safety, or it is better in terms of efficacy than the drugs available in the market. So can you give me a fast track designation? And the FDA says yes. And what are the benefits which come with the fast track designation? The FDA will therefore review your package much more closely. They will handhold and support you in the end of phase 1 meetings or the end of phase 2 meetings. They will work with you on the protocol design. They will work with you on the endpoints, and they'll basically be overseeing your program. And that's the advantage you get the FDA, because the moment they handhold you and work with you, they will ensure that the drug reaches the market, the drug is really worth its salt, right? So that's how the fast track designation works. In a breakthrough designation, primarily you're looking at a drug which is really breaking all efficacy barriers. So you have a certain compound in the market for a particular disease, but your early data is so very strong that it is a significant improvement over existing therapy. You can apply for a breakthrough designation. Right? And a breakthrough designation basically will give you all the advantages of a fast track designation, right? Because you've got a drug which really is very, very superior than the drugs on the existing market, right? For an accelerated approval, I mean, it's pretty much mirrors a fast track designation, breakthrough designation. An accelerated approval again is given more in a fast track designation, and you basically can get away with surrogate endpoints. You don't need a comparator study. You can do a single arm study. You can have a surrogate endpoint like a overall response rate instead of an overall survival. So again, an accelerated approval can apply both to a breakthrough designation and a fast track designation. But you get away with single arm studies. You can get away with surrogate endpoints, because these are being tested in cancers which don't have too many therapies available, or the therapy which you have is significantly better than the available therapy. So you get an accelerated approval as well in such conditions. Mind you, an accelerated approval, the surrogate endpoint is written on the label of the product. So your product label will say that this product is being used to treat relapsed refractory so-and-so cancer, and this approval is under accelerated approval. So they will always mention that in the label, because your accelerated approval is based upon a surrogate endpoint. You still have to do, after you get the permission to market the product, you still have to do studies which will lead you to a full approval, which means that you still have to conduct clinical trials which will evaluate overall survival as the endpoint, right? And the FDA will say that, okay, we're giving you a marketing authorization right now based upon an accelerated approval, but you continue to do studies which will eventually give me the overall survival data, right? So the accelerated approval comes with a caveat, but you're still able to reach the market faster. When you look at priority review, we're looking at typically a regulator looking at your data with more vigor and on an expedited basis, right? So if you look at the standard PDUFA, the Prescription Drug User-Fee Act of the US FDA, right? There are certain timelines by which the FDA will review the application package or the new drug application when you file for your drug. So the standard timelines typically are about 10 months, right? So once your whole package is submitted, the FDA will take anywhere between 10 to 12 months as a standard review time for them to review the whole package and then decide whether they can approve the drug or not, right? In a priority review, that timeline is crunched to 6 months, right? So priority review again is given only to those drugs which are meeting an unmet medical need, which have a fast track designation or a breakthrough designation. So you have to have some advantage of your drug, which really is able to sort of overcome the lacune of an existing therapy, or a therapy for which, or a disease for which there is no therapy available or no good therapies available. You can apply for a priority review, and that crunches your timeline to about half, to about 6 months. So these are some of the mechanisms by which the FDA pushes and promotes early development by pharma companies, right, or biotech companies, so that they're able to get the drug development cycle much more faster and get the drug to the market much more quickly. This is again an amazing thing which has happened in 2000, in the late 2017s, 18s, which is called the FDA's RTO, Real-Time Oncology Review Initiative. This is again a path-breaking sort of initiative by the FDA, which probably no regulator has really done this in the world, right? So this was launched in June 2018. And these are some of the drugs. If you look at some of the drugs here and look at the timelines, and then the timeline below that drug, right? For example, alpelisib of Novartis, it took 5.3 months to get approved post-filing, right? Like I said, the standard timeline is 10 to 12 months. A priority review is 6 months. But this is again before a priority review. Calquence, acalabrutinib from AstraZeneca, got an approval in about 2 months. Etcetras from Seattle Genetics, brentuximab vedotin, got an approval in 11 days. But remember, brentuximab vedotin was already approved for another cancer. This was a supplemental new drug application. So when you have a supplemental new drug application, you're basically increasing your label, which means that Etcetras was approved for classical Hodgkin's lymphoma, and then that was approved for many years now. They also did studies in peripheral T-cell lymphoma, and then they applied for peripheral T-cell lymphoma application via the RTO initiative. Because the FDA already knew a lot about brentuximab for Hodgkin's lymphoma, they were able to review the whole package and make a decision in 11 days to approve Etcetras for peripheral T-cell lymphoma. If you look at Zospasta, which is gilteritinib for acute myeloid leukemia, in 3.2 months, they got an approval, right? So we're looking at an extremely crunch timeline by the FDA when it comes to this real-time oncology review. And this basically happens because as and when you produce the late-stage development data, you can submit that to the FDA in real time. You don't have to really wait for all your documents to be ready to make a submission to the FDA. The FDA says that give me efficacy, safety, clinical pharmacology, CMC manufacturing control data as and when you feel they are ready. So essentially the FDA is basically asking you to submit the data as and when it is ready, because they can start reviewing it then and there, right? So by the time you submit the last data package, the FDA has already reviewed the first and second package. And because of which, when you submit the last package to the FDA, that's probably the only package left for review for them to make a decision. And because you've seen packages 1 to 4, if 5th is your last package, they pretty much have an idea that seeing the 4 packages, this drug is good to be approved. So the 5th package is really reviewed very, very quickly, and you can get an approval within about whatever time frame you see here, 3 months, 2 months, 5 months, 5 months. So this is the way the FDA will encourage companies to submit data as and when the data comes through, and not wait for that full data package to be submitted. And that's because the RTO initiative of the FDA, which has really helped a lot of companies and a lot of patients eventually, because these drugs, when they get approved, they can really be marketed very quickly and reach the patients who really need this compound. For example, a drug like Etcetras for PTCL. PTCL is an extremely dangerous disease. I mean, the overall survival, I told you, for 5 years is less than 30%. Gilteritinib for acute myeloid leukemia, and this is typically for a mutation, of the FLT3 mutation. Patients with AML again progress very rapidly. The average overall survival is less than 2 months. 20% at the end of 2 years, right? So these patients need these drugs faster, and these initiatives by the FDA really help these drugs to go through development cycle quickly and reach the patient. Yeah. And this is just to give you a perspective of the comparative timelines of the FDA versus the EMA. I'm just checking my watch so that I'm within the time frame. If you really look at the comparison between the FDA and the EMA when it comes to oncology regulatory approvals, you can see that the FDA is probably taking half the time which the EMA takes. Now, there is both, there are two schools of thought here. One is, of course, the FDA is amazing here because the FDA is really reviewing data very, very quickly. They're looking at they're keeping the patient in mind eventually, because the FDA is eventually responsible for the health of American citizens, right? So they believe that how quickly can you get your drug to the market so it reaches the patient eventually, right? So the FDA really has all these initiatives put in place which helps them give the approval in half the time which the EMA gives, right? The other school of thought is that is FDA looking through data so very rapidly that are we having quality issues, right? So there have been some trends which have been observed where after the product is approved by the FDA through its accelerated route or through its RT or a program, there could be a lot of safety events happening. For example, more adverse effects happening with the drug when it's launched in the market. So the FDA has to approve, change the label, right? So the FDA will tell the company, rechange your label. They will probably add a black box warning if there is a serious toxicity coming in. So if there's example, during the quick review, there was no toxicity seen, but when you launch the product in the market, right, there was a lot of toxicities which was seen, and therefore the FDA will say, okay, please relabel your prescribing information, and there'll probably be a black box warning. So the other school of thought says that maybe the FDA is looking at data too very quickly, and they may miss out on some things, and therefore that missed out thing goes to the market, and the patient will bear the brunt, right? So two schools of thought, but I think overwhelmingly there is one school of thought which says that the FDA is the most proactive and forward-looking agency when it comes to development of oncology products as compared to the EMA or any other agency in the world. And I'll probably end my talk with a couple of case studies, and this is just to give you a perspective of what I talked about, right? This case study is primarily for the first one is for the targeted therapy for non-Hodgkin's lymphoma, which is basically a hematological malignancy, which is a malignancy of the lymphoid organs, right? So it's a therapy for non-Hodgkin's lymphoma. It's a aggressive tumor, like I said, with a 5-year overall survival of less than 30%. The therapies are available, right? But I mean, pretty much the frontline therapy is a cocktail of chemo agents, right? So it's a chemo cocktail which is given as a frontline therapy. But pretty much a lot of patients will relapse, right? After about 18 months, after about a year, the disease will come back because the chemo would have failed. Or some patients are refractory. They will not respond to chemo. I mean, normally for this disease, you have a 6-cycle chemo, right? After about 2 cycles also, there is no change in the tumor measurements. The tumors still remain the same. The patient is still sick. That's a refractory patient. So obviously, we're looking at a very refractory disease. And when the first line fails, there are some drugs which are approved as second line agents, right? So after the first line of therapy, which is chemo, fails, there are some drugs approved for relapsed refractory conditions. Even in those conditions, I mean, even for those drugs, your response rate is very, very low. It's about 25%, which means that 1 in 4 patients really show a response to that second line therapy. So there's a company which developed a drug, right? And in the phase 1 study of the expansion cohort, remember the basket trial which I talked about, in one of the tumor types, which happens to be this non-Hodgkin's lymphoma, it showed at a very early stage of phase 1 a very good response of 45%, right? A 45% response rate, which is actually much more than what is there in the current available market, which is 25%. So at the early development stage itself, we've seen a 45% response rate as well as a 25% available in the market. The company can actually apply for a fast track designation because it meets the criteria of a fast track designation because the disease is a very difficult to treat disease. The disease doesn't have therapies which really have too much of hope. It's a 25% response rate. You can apply for a fast track designation. And then once you apply for a fast track designation, you can negotiate with the FDA for an accelerated approval, which means that you can now use a surrogate endpoint and not a full endpoint. So you can basically use a surrogate endpoint like an ORR as against overall survival, and then negotiate with the FDA for a single arm, say 70, 80 patient study in this indication and get an approval right from your expansion cohort, right? And that's the beauty of how you can use the available regulation, right, to expedite drug development in a rare condition like this non-Hodgkin's lymphoma. Yeah. I'll give you a case study too, right? And this case study again talks about an overall aspect of how do you really fast track in a solid tumor situation, right? So again, I mean, looking at a therapy which is for tumor expressing a certain mutation, right? For example, as you know, there are multiple gene mutations which happen in multiple cancers, and some of the gene mutations specifically, right, are known for certain cancer types, like a BRCA mutation in breast cancer or a BRCA mutation in ovarian cancer. It's a loss of function mutation. So basically, by that mutation, there's a loss of function of that gene, and because of the loss of function of that gene, the genomic instability happens and the tumor starts growing, right? So it's a targeted therapy for a specific mutation loss. Again, going back to our basket design, I will select patients with multiple tumor types in which this mutation is present, right? So you can have breast cancer, you can have an ovarian cancer, I can have a pancreatic cancer, I can have a lung cancer, a non-small cell lung cancer, and NSCLC. When this was done, you see that one arm, which is NSCLC, right, is recruiting faster, right? A lot of patients are recruiting faster, and you see that the drug is showing a result, right? And once you see the drug showing a result, what I can see is that the drug result is so very fantastic that it probably qualifies for a breakthrough designation, right? So if you look at breakthrough designation, remember the definition I told you. Breakthrough designation is something which has an efficacy which is much higher than what is existing, right? So if this drug in non-small cell lung cancer for that mutational type, it shows a efficacy which is much more superior than what is available in the market, you can apply for a breakthrough designation. And with that breakthrough designation, you can do a comparative study with the standard of care, right, with a superiority endpoint. So essentially, you will design that study, that same cohort will tease out, branch that out into your study compound and your standard of care, and then do a comparative design, and you get a full approval with overall survival as the endpoint, right? So this is again an example where you can use a regulation of a breakthrough designation for a compound which shows promise much more better than what is there in the market, and really fast track your drug development and get a full approval with overall survival as the primary endpoint. So this is the other case study which I was going to talk about, right? So I think I've probably overshot my time, but this is what I really want to talk at a very large level in terms of the overall oncology development regulatory landscape, and how the oncology space can basically be taken as an area where drug development can be expedited, and that you really can do the drug development in a much more quicker way. Okay, so thank you everybody for your patient listening, and I think I can take questions now, and if anybody wants to know more details, they can reach out to me. But thank you for your patient visit.
Yeah. So if any of you have questions, you can ask. We'll take maybe probably one or two questions. Okay, so it looks like there are no questions. Thank you, Dr. Nair, for your wonderful talk on regulatory strategies, oncology, and development. Thank you.
Thank you.
So we'll move to the next speaker, and the last talk of this session. I'm happy to introduce Mr. Jimesh Shah. So Jimesh Shah, working as a senior manager in some pharmaceutical industries limited, R&D center since 2009. Previously, he was working in regulatory affairs development in Torrent Research Center, Gandhinagar, for 2 years. Jimesh was working in quality assurance department as an initial career in Intas Pharmaceuticals, Ahmedabad. Jimesh is Bachelor of Pharmacy and MBA in Operations Management by Education. He has total 15 years of research and development experience for regulatory affairs and quality assurance. Now, over to you, Jimesh, for the talk.
Yeah. I am audible?
Yeah, very much.
Yeah, thanks, Mr. Gaikwad. Firstly, very much thank you to BITS Pilani team that they have encouraged all the participants and give the opportunity for the regulatory and the patent parts. So I'm very delightful that I'm providing the general requirement of the regulatory, that is for the US and the Europe part. So just I will share the presentation. Is it visible?
Yeah, it's visible. Just make it full screen, full mode of presentation. here in the bottom you can make the full mode. Yeah. Now it's visible now. Yeah. Please go ahead.
Yeah, thank you. So just my topic is that what are the similarity and difference in the regulatory approval procedures for the US and the Europe. So here we are discussing about the generics. What are the generics that we are filing through the industry? But a lot of people doesn't know what are the main difference and what are the main similarity that we have to file the product in the US and the Europe. So basic question is what are the generic drugs? So we know that it is a very safe and effective alternative to the brand name prescriptions. It will help both consumer and the government reduce the cost prescription of drug. And as already FDA and the EMA, that is the Europe authority, says that when a generic drug product is approved, it has met rigorous standard established by the FDA with respect to the identity, strength, quality, purity, and the potency, right? So generally industry are very keen to develop the generic drug compared to the reference product drug. Yeah, reference product drug is very costly in the market. So to cater that, industry always effective and provide the generic drug development part. So what are the requirement of the generic drug? Here we have the if same active ingredient, same route of administration, same dosage form, same strength, same condition of use, and compared to the reference listed drug, that is the innovator drug, which is the brand name product. So these are the requirement for the generic drug. What it means by same active ingredient? It is nothing but the API. Route of administration and dosage form, it means it's a tablet, it's an oral route. What about the strength? Strength, if it is a 5 mg innovator, we have also developed the 5 mg. Same condition of use and compared to the reference product drug. So these are the basic requirement of the generic drug that we have to cater. Now, this is the main question. What is the regulatory affairs and what is the dossier for regulatory submission? So everyone know the regulatory affairs, what are the dossiers, what are the requirement, but what is the exact means by regulatory affairs? Regulatory affairs means is nothing, but it is a profession which act as an interface between the pharmaceutical industry and the drug regulatory authority across the world, and it is mainly involved in the registration of the drug product and device respective country prior to the marketing. It means that if I want to file some product in the US or Europe, I have to follow the guideline of that particular country, and for that, industry should contact with that authority and should find which are the guidance that I have to follow. This is the regulatory affairs exactly doing. And the dossier or the document which we have provided to authority as per the ICH format, it is called as a dossier. So registration dossier of the pharmaceutical product is a document that contains all the technical data, which is administrative, quality, non-clinical, clinical, which is approved and registered, marketed in a country. So dossier is nothing but combination of all administrative, quality, that is the CMC, non-clinical data and the clinical data. When all the documents come together, it becomes a dossier. So these are the basic challenge. We can say that when we file the dossier, administrative, quality, non-clinical and clinical data should be available at a one time. So these are the definition of regulatory affairs, and this is the dossier requirement. Now, I have divided some of the pharmaceutical classification of this market, which comes under the regulated, semi-regulated and the non-regulated market. Regulated market, as earlier we also saw, that which is a very stringent, that follow the ICH and very stringent document requirement. Example like US, Europe, Canada, Australia, Japan, which are the very regulated, highly regulated market. Comes to the second point, that is a semi-regulated market. country-specific product registration guidelines are followed. That is the Asian country, Russia, Brazil, China, which are some of the semi-regulated market. Non-regulated market comes some of the African countries that minimum documentation is required for drug approval process. So what are the basic three differences that US, Europe, Canada, Australia, they are more follow the ICH and the guidance which we do not deviate any time. Semi-regulated market, they have the country registration guideline and somehow they follow the US, Europe, Canada and Australia guidance. And African country have less stringent requirement that we get the approval very early stage. So these are the main classification of the pharmaceutical market. Now I'm coming to the US FDA review process. Basically, there are the ANDA and the NDA. There are the two processes when we file in the USA. So what are the basic difference between the NDA requirement and the ANDA requirement? It's nothing but it's a brand name of the drug in the NDA. Animal study and the clinical study basically are the main requirement for the NDA. It is a new drug application. ANDA is already, we know, abbreviated new drug application, innovator is available. So most of the data of the ANDA, we have to follow the chemistry, manufacturing, controlling, label, testing, and bioequivalence study. That is the human study that we have done. These are the requirement of the ANDA. So basically, US FDA review process is a simple process and they have the two parts. Now, what are the generic drug review process? How the FDA will approve our dossier. So these are the main flowchart that FDA follow while giving the approval. So first we can start with the applicant. Applicant is nothing but the industry. So industry can file the ANDA, then it comes under the application review. There are the certain criteria that we have to follow while filing the application. If there is a gap between the application, it comes under the refuse to receive letter. That is the right side I have mentioned. That is called RTR letter. It means that agency has denied your application and you have to refile your procedure. If it is acceptable and complete, then it comes to the four part. It is first part is the request for plant inspection. That is the GMP part if it is required. Second is chemistry and micro review. Third one is the labeling part and fourth is the bioequivalence review. So after acceptance, there are the four category that FDA will review separately and separately they will follow the pre-approval inspection, chemical micro okay, labeling, bioequivalence. So these are the four and all the approval will come and then they give the approval of the ANDA. So these are the basically 10-month review process while giving the review. So I have also mentioned here the 10-month review cycle from the FDA side. From they have product development meeting to the pre-submission meeting through the ANDA submission, filing determination, DRL that is the deficiency, mid-review cycle meeting and the regulatory action. So these are the basically 10-month cycle for the FDA for approval of the ANDA. So these are the story about the US FDA. Now it comes to the Europe market. Europe is very interesting 4 type of procedure while filing the dossier. So basically there are the 4 centralized procedure, decentralized procedure, mutual recognition procedure and the national procedure. It is nothing but centralized procedure we can file the product throughout the European Union. Decentralized procedure we have to select the country and we have to file the procedure. MRP the product which is already approved in any one of the country we can run as a mutual recognition procedure. And national procedure is nothing, we can only file the product in the single country. So these are the beauty of the European procedure, that there are the 4 procedure. While in the US it is already governed by the only FDA, but here it is a different 4 type of procedure. Also in the Europe, legal basis of application is very important. What is the legal basis of application? It is nothing but when the innovator is approved, you have to follow that innovator and your product is the similar. If it is not similar, it is not considered under the generic application. In the US FDA, it is already mentioned RLD and RS on the FDA side, but in the Europe, it is not available. So while we filing the procedure, legal basis of application, we have to confirm with the authority before filing, and based on that, we have to decide it is a generic application or the hybrid application. So these are the few legal basis of application. Article 8.3 means it is a full application. Article 10, it comes under the generic, hybrid or biological application. And there are few 10A, 10B and 10C, well-established fixed combination and the informed consent application. So these are the legal basis of application. That is also difference from the US FDA part. Now approval timeline in the Europe. It is also very interesting. If we file the procedure with the DCP, they have the different time. If we file with the CP also, they have the different time. If with the MRP also, they have the different time, and the national also, they have the different time. So we can start with the DCP part. If we file the DCP application, it's a generic application take 10 to 15 months and hybrid take 12 to 8 months. About the CP part 10 to 15 and hybrid 12 to 18. MRP, it's a very little because already one country is already approved there. So 30 day to 120 and maybe 30 to 180. And national, I think there is no timeline, but approximately I have put 10 to 48 months and 12 to 60 months. So these are the approval timeline for Europe, which is based on the clock stop that we have taken generally. Now, how we file the dossier. So these are the common requirement for any Europe or the US FDA. What we first do, we have files are attached as per the module in the ECTD backbone using the software. We can use any software which is available in the market, which is US FDA and Europe compliance. Then we compile the full dossier. Then enter document or hyperlinking the compiled folder. We then synchronize and assimilate the hyperlinking. We can final compilation of this dossier. Then we validate all the ECTD structure using the software. We can then verify of all attached file and complete sequence, and then finally publish the sequence to the ESG gateway. So these are the general procedure that we follow for the filing of the dossier. Now, what are the principal difference between US FDA and the Europe? US FDA is only single agency. So the dossier will be reviewed by only one agency. Europe have the multiple agency, EMA, CHMP and the National Health Agency. So they have the different opinion while we file the dossier. Here US FDA only one registration process. Europe, they have the multiple registration process. It is a centralized, decentralized, mutual and national as we have discussed. US FDA, yeah, TSC-BSc study is most probably not required. Europe, we have required. US FDA Braille code for the blind people. Braille code is not required during the labeling. Europe, yes, most probably they have insisted most of the country Braille code for the labeling part. Yeah, after approval, US FDA have the procedure of the PASS, CB30, CB and the annual report. It means that after approval, if we want to change something, we have to follow the procedure of the post-approval cycle. And also Europe have the different procedure, that is a type 1A, type 1B and the type 2 variation. So these are the main difference, principal difference between the US FDA and the European procedure. What is the principal similarity between FDA and Europe? First is 3 batches required at the time of filing. So what it means that 3 batches data? It means that if we want to file the dossier to the US FDA or the Europe, we want 3 batches data, means we have to manufacture 3 batches at the time of filing and we have to gather all data and compile the dossier. So these are the requirement both for the US FDA and the Europe. Europe sometime if it is a IR tablet, means it is an immediate release dosage form, sometimes they accept the 2 batches data also. But it depends on the API stability, that is the drug substance stability. Second point, 6 month accelerated and room temperature data required at the time of filing. What it means that at the time of filing, accelerated and the CRD, that is the 2560 and 4075 stability data required, means minimum 6 month data required at the time of filing. Third point, ICH guideline to be followed for both the country. Both the country have the own guidance as well as ICH guideline. So we have to follow the guideline rigorously. Fourth point is the product development required to file the dossier. If we want to file the dossier, we have to provide the product development report along with the innovator product. So first the formulation team developed our internal product based on the innovator is available. So there are the QBD, that is the quality by design approach we have to follow in the product development report, and we have to finalize the product development report to fulfill the dossier requirement. Both the country have the ECTD, that is the electronic common technical document structure. Yeah, except module 1. Module 1 is nothing but the administrative part, that the US has own administrative part, Europe has its own administrative part, because Europe has its own languages. So except module 1, module 2 to module 5 requirement are almost similar while we compiling the dossier. Both the countries have the applications fees also. Yeah, so when we file the dossier, both the country has its own applications fee. But in the US, there is a one that is a refuse to receive file. That is the rejection of the dossier is possible, while in the Europe, rejection of dossier is not possible. So there are the main difference between the US and the Europe. Both the country have the CC and the scientific advice procedure. It means that if you want to ask something to the agency by the control correspondence or the scientific advice, you can check with the agency this is okay or not. Both the countries require the bio study and the resolution study. Based on the dosage form, yes, it is a requirement of the bioequivalence and the resolution study. So these are the main principal similarity between the FDA and the Europe. Thank you. So I am more rigorous about the question answer.
Thank you, Mr. Shah. Now the session is open for questions. Anybody has questions, they can ask. Okay, so it seems there are no questions. Thank you, Mr. Shah, for giving such a wonderful talk in the comparison between European Union and the US FDA comparison between dossier, etc. Thank you very much.
Thank you, sir. Thank you.
So this brings us to the end of this session, as well as this one-day conference on intellectual property rights and the regulatory affairs. Now I request Professor Hemant Jadhav, the head of the pharmacy department, to give a concluding remarks and to end the one-day conference. Thank you. So Professor Hemant, over to you.
Good evening, everyone. So, because of the pandemic, we could not have conferences on campus. We wanted these conferences to be here, wherein the expert speakers can come here and discuss with our students, not only during the session, but off the session also, because in any conference, the off the session discussions are very fruitful and very important also towards the young minds which are coming up in this field. Nonetheless, when the lockdown was extending, then we thought that why not have some at least discussions and online sessions for our benefit of our students, and make these sessions available for everyone, not only to BITS Pilani, but the other stakeholders also. So with this intention, we had organized the series of conferences. We had 3 conferences. October 30, 31st, we had a conference on the computer-grade drug design. The second was on the drug discovery and delivery on November 6 and 7. and this is the third virtual conference on the regulatory affairs as well as intellectual property rights related to pharmaceuticals. So, as today morning Director Sir mentioned, Professor Barai mentioned, that we began with the design, then the discovery part, then delivery, and finally concluded with the regulatory aspects. So, I hope that this series is complete, and what we have done is for benefit of everyone else, we have uploaded these lectures on YouTube, so that at any given point, if someone wants to review, someone wants to revisit, or someone wants to share, they are available, and that is the best use of internet we can have, wherein we can share the information, right? Not in the real time, but as well as the off the time limits, beyond the conference also this can be made accessible. So in this series, we had speakers from academia as well as from industry as well as from various other organizations, non-governmental organizations. We also had a good support of our faculty members, research scholars, as well as the participants who have joined these sessions from not only from India, also from the other countries as well. In particular, in this conference, we did not plan it for 2 days because our previous experience was 2 days becomes too lengthy, and to maintain the crispness, we had 6 speakers, all from industry, touching upon various aspects of regulatory as well as the intellectual property right area. Right from Dr. Patnaik from Teva to the last speaker, Mr. Jimesha, right? All have given different views of the regulatory aspect. In fact, when we were having the lecture of Dr. Nair, who spoke in great detail about the oncology clinical development, we have a course called clinical research, and this lecture would be much useful to them as well. Course in the sense a subject, in which language we call it course, whereas others they call it subject. So we have a subject called clinical research, and that discussion will be very much helpful. Farheen, Dr. Patnaik, Dr. Mr. Jimesh, and Dr. Mishra, whatever they spoke about, that is helpful for our students who are learning the course, that is subject on quality assurance and regulatory affairs. So, in a way, this conference is useful to the newly joined master students, to those students who have done these courses, to the outside participants also, because they are getting a different view. So, in a way, I consider this series of 3 conferences a success in that perspective, and all the lectures are online, so it is very good medium for students to review those topics as well. So I thank all the speakers for today, Dr. Patnaik, Dr. Mishra, Mr. Shah, Dr. Nair, and Dr. Pinaki, and Dr. Farheen. We are very much thankful for agreeing to speak on a short note. In fact, we started planning only last week, and all of you very kindly agreed to speak, and we immediately prepared the brochure and shared with everyone else. We had good participation also, although online we can see here there are 74 participants, but we know that in Google, in YouTube, there are some participants who are viewing, and some may be a little relaxed. They would be thinking that, okay, why not revise it later instead of seeing it today itself. So that's the benefit of internet, and they are using it the most. So I thank all the speakers who have taken time out of their busy schedule and presented the information for the benefit of the science as well as the society. I thank the faculty members, Dr. Gaikwad, Dr. Deepak, Dr. Anil Jindal, Dr. Murli Pandey, because they brought in their contacts on a short notice. These contacts, these speakers were made available because of their efforts. So I thank them also. I thank other faculty members as well for their support. And the research scholars, master students, for being patient and they are attending even without registration also. So they all have been very patient because we send them link the last moment. Yesterday itself, I sent the students this link for this conference, but participants we sent day before yesterday. So the students also I should thank, and we are very much happy with the outcome of this series of conferences. So for the benefit of students, maybe as a conference or maybe as guest lectures, we would like to continue this session. Probably next month is the comprehensive part. So next to next month in January, February sometime, if time permits, we would like to arrange more such interactions with the industry people as well as renowned academics, and we will make it available to the outside participants also. In fact, all the speakers, I'm thankful, and we once the lockdown opens or once the COVID situation subsides, and when we have a free movement, then we would like the speakers to come to the campus, see our campus, motivate our students, interact with us, and as in the beginning, Director Sir mentioned that yes, we are open to collaborations, to outside participants also, to anyone. We are open for collaborations. So thank you very much. One and all, I should hope to see you again when we arrange such sessions in the future. Thank you. Goodbye. Good day.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.